David Okello and I have worked together a lot on this work, and David Dr. Okello is a groundnut breeder in Uganda. And I've worked with him and he's carried the load. I've just kind of been around to give support and encouragement and type of things. At any rate, I would like to thank David Jordan and the program committee for giving us the opportunity, the invitation to speak and bring everybody up to snuff on groundnut rosette disease. I'll talk a little bit about disease, past, present, and future. So what is groundnut rosette disease and why is it important? Well, it's the most destructive viral disease on groundnut in Sub-Saharan Africa. Can be arguably, it's the most important pathogen on groundnut in Sub-Saharan Africa. The disease can cause significant economic losses, and in young plants before flowering, these can be up to 100% yield losses. And for smallholder groundnut farmers, this routinely can jeopardize food security and their livelihoods. Groundnut rosette disease can have a long-term debilitating impact on production. This isn't so much true now as it was in the past, but in the past, following epidemics, farmers would decrease production just to avoid future risk. And to productively grow groundnut in some regions of Sub-Saharan Africa, and what I'll be talking about today applies as much to Uganda as any place else. David Okello, Dr. Okello is a breeder at the National Agricultural Research Organization in Uganda and stationed at the National Semi-Arid Resources Research Institute in Serere, where he does most of his work. So a lot of what I will say today is from our experiences in Uganda, obviously then. So to productively grow groundnut in some regions, such as Uganda, requires groundnut rosette disease resistance. So just as in context, put everything in context, if you look at worldwide production of groundnut, about 34% comes out of Africa. And if you look at grain legume production in Africa, the largest percentage is with groundnuts with about 16 million tons, metric tons, which is about double the second closest with cowpea. Now, groundnut is obviously beneficial for a number of reasons. Everyone knows its importance, its contribution to dietary protein, oil, and micronutrients, but it's also very beneficial also as it provides a cash crop and income to small shareholder farmers and is important in replenishing soil nitrogen. So has many important aspects. And in Uganda, another important aspect of groundnut is it's part of the culture. So it's the cultivars that are grown are very important. Seed cover color is very important. Just numerous things that makes it part of the culture and ground in Uganda. And so the breeding program addresses many of those aspects, as you'll see later. Groundnut rosette disease history. It was introduced into Sub-Saharan Africa. Groundnut was from South America and by the Portuguese in the 1600s. The disease itself was first reported in Tanzania in 1907. The disease is endemic to Sub-Saharan Africa and its offshore islands. So to my knowledge, it's never been found outside of Sub-Saharan Africa. There were claims in the past, but they were eventually identified as other viruses that caused symptoms that might have been very similar. And by the 1960s, it was very clear that more than one virus was associated with the disease, which brings up the etiology of the disease. It's caused by a complex of 2 viruses and a satellite RNA. Now, satellite RNA is a subviral agent, parasitic RNA that requires co-infection with a helper virus to replicate the satellite RNA. The satellite RNA has no similarity to the virus, and it does not encode for proteins. The disease is transmitted by the groundnut or the cowpea aphid, Aphis craccivora, which feeds on a wide variety of plants, many in the Fabaceae, which includes the legumes. The aphid is an invasive species of cosmopolitan distribution, which is another way of saying it's worldwide and is found in any environment where it can thrive. And it's the most important pest of groundnut in the tropics. Now, the groundnut rosette disease components, the 2 viruses and the satellite, are shown here. This is a very simplified version of the genomes of these 2 viruses, just so I increase clarity in this particular circumstance. The first virus component is Groundnut Rosette assistor virus. It's a ludio from the Ludoviridae, which doesn't mean much to most of you. The important aspect of that is that it's limited to the phloem of plants. It has both of these viruses are plus single-stranded RNAs, which and all that means is that when they're introduced into a healthy cell, they come apart. The messenger RNA is immediately translated to give the polymerase proteins and the proteins involved in replication at the 5' encoded at the 5' end of the genomes. The GRAV, here again, Ludovirius, phloem limited, has a coat protein, and a low percentage of that time when that coat protein RNAs run off and translated into coat protein, a low percentage of that time, that messenger RNA when it's read, it'll read through the stop codon to give a C-terminal addition to that protein, which is required for aphid transmission, or the AT. So you have the coat protein made small amounts of CPAT. The Groundnut Rosette virus, it's an Umbravirus in the Tombosviridae family. It does not encode a coat protein. The satellite RNA of GRV is required for symptoms, the symptoms being 2 major groups. Chlorotic symptoms are green symptoms, mosaic. I'll show those here shortly. The key to this whole affair is that Groundnut Rosette virus and the satellite are not limited to the phloem. They will move into the mesophyll tissue of the leaves and propagate. And so you get large amounts. However, for them to be moved by aphids, they have to get the virus and the satellite that replicate in the phloem will become encapsulated into the coat protein of GRAV. So if you look to the right, GRAV provides the protein for encapsulation of GRV and the satellite of GRV. They have to be packaged together. GRV is the helper virus which replicates the satellite then. And so this continues just to want to do this. So it's a fairly complex interaction between the 2 viruses and the satellite. If you look at symptoms, I mentioned Rosette's chlorotic symptoms as seen on the top 2 panels, green, Rosette seen on the bottom 2. This thing that's always fascinated me about the green rosette is that you'll notice that the green color is much darker than the plant to the left of it, which we're assuming is uninfected or a healthy plant at this point in time. In the field, late in the season, when you have a lot of leaf spot, the green rosette is much more easily identified, where the chlorotic kind of fades into the background of the leaf spot, depending on how bad the leaf spot disease is in the field. The aphids are shown on the left, craccivora. You'll see nymphs on the bottom. On the top, you see the black-winged adult aphids. They're females, and they are the ones that transmit the virus from plant to plant. Aphid acquisition of GRD, groundnut rosette disease agents following phloem feeding. And these are the transmission scenarios. And this is where the complexity comes in again. If the aphid picks up, it can pick up both the components or each one individually. If it picks up both components and it transmits both components to a healthy plant, then you get disease transmission and disease spread. And you'll see that in this example on the left here. If it transmits just GRV satellite RNA, or if it transmits just the GRAV, or if it only picks those up and transmits them, then you have two different scenarios, slightly different. GRAV alone, you get no disease transmission because you got no GRV or satellite. If it transmits just the complex, just the particle of GRV and the satellite RNA in the particular plant, the healthy plant that receives this, you can get an infection showing groundnut rosette disease symptoms, but it can't be transmitted from that plant because there is no GRAV. So that becomes a dead end. So the important aspect of the transmission scenario is when both components get transmitted to a healthy plant. Just to bring this all together now, distribution of groundnut aphids and groundnut rosette disease. Again, groundnut rosette disease is endemic to Sub-Saharan Africa. You'll see major groundnut growing regions in Asia, Sub-Saharan Africa, in the US, some in South America. This is an older slide, and the distribution of rosette disease and the major groundnut growing areas would stay very similar. The aspect that would change on this is that you'd see much more yellow on this world map to indicate how wide the range of craccivores, Aphis craccivores. It's probably minimized here, and it's probably much more extensive than seen on this slide. If you look at, moving on to looking at tactics for management and control of groundnut rosette disease, these are very similar to what are used for a lot of pathogens. The most important aspect, as far as we're concerned, is the resistant cultivars, and that's generating new germplasm lines with resistant groundnut rosette disease resistance. This is the most accepting and practical in developing countries and Sub-Saharan Africa. A lot of these tactics depend on resources that the farmers have. So in developed countries, the resources are more plentiful. Chemical approaches can be taken, more cultural practices can be taken along with resistant cultivars. In developing countries, we're working more from the resistant cultivar aspect. However, chemical insecticides do work, and especially useful in young plants. They're more susceptible to disease before flowering. Cultural practices, for example, planting early in the growing season to avoid high aphid populations has been shown to be to work quite well, as well as cereal intercropping and border cropping to minimize aphid movement and population size. And both of these were touched on by Albert and tomato spotted wilt virus, as was planting density. Optimal planting density that results in rapid ground coverage minimizes attracting aphids. Typically seen when you see it in the field, many times you'll see it at the end of the row where you have areas of the soil that aren't covered with foliage, which would attract aphids then. and might introduce the disease into the field from the end of the rows. But planting density is an important aspect and is used. And the last one, roguing, some people might use that, but it probably doesn't work very well, because usually when you see the disease symptoms in a plant, the insects that carry the virus have moved on to additional plants that have been feeding and passing the disease on. And so as far as groundnut rosette disease goes in Sub-Saharan Africa, the most important aspect of talking about from the management aspect would be the resistant cultivars then. Resistance comes in a couple of different types, but there's so much more to be learned about this. Host plant resistance, groundnut rosette disease resistance, was initially identified in land races from late maturing Virginia types from West Africa. Resistance identified in these lines was directed towards groundnut rosette virus, the helper virus for the satellite, if you remember. Therefore, it's effective against both green and chlorotic symptom types, since if you don't have GRV, you don't have the satellite. The resistance is governed by two independent recessive genes. And while there's numerous sources of resistance to GRV that have been supposedly reported in the literature, it's thought that most of these forms, the majority of these resistances originate from the resistant lines from West Africa. To date, there's no details available on how the resistance mechanism functions, which is unfortunate and would be helpful. And hopefully this is an aspect in the future that needs to be addressed in a big way. There is also vector resistance. However, this resistance is broken under high aphid pressure. This resistance is available as a single recessive gene, and it's thought to induce a hypersensitive response at the site, aphid feeding site, which minimizes spread and localizes the virus. Groundnut varieties and new replacements. And so, as I mentioned, our groundnut rosette disease resistant cultivars have been the way to deal with the disease, the epidemics in Africa in the past, or disease of production losses in the past. In Uganda, the number of old varieties were the SARNet 1 through 4. These were replaced by 2010 by a number of new varieties, and most of these all have value-added traits. And all of them have access or have groundnut rosette disease resistance. They have leaf spot resistance, most of them do. And of course, their yield levels are much higher than the old varieties that they replaced. SARNet 4T was one particular one that was used heavily for a while, and that has that vector resistance that I mentioned to you. SARNet 2, another popular variety, does not, or SARNet 1R, another popular variety early on, does not have GRD resistance. So, and the varieties become in two forms, with a red seed color of the R or a tan seed color T. And I mentioned to you that groundnut is important to the culture of Uganda. Certain regions like red seed color, certain regions like tan, and it's very important to them. And David usually breeds cultivars of both red seed, both color, both red and tan. Varieties, the various Spanish and Virginia, with some of them being PIs like the early ones, the SARNet 1 through 4. More recent varieties, that new and upcoming varieties, NarrowNet 1R and 2T, here again, red and tan seed colors. These two particular lines are pretty exciting, and not only because they have high yield, which is obviously important, and the major aspect of the breeding program throughout is to increase yield, but due to their early maturing dates of 75 to 85 days, which makes them more drought resistant. obviously, if rains stop, depending on when, all of this is rain-fed groundnut in Sub-Saharan Africa. So getting crops, the earlier you can get crops many times will avoid a drought situation later when you might have yield losses because the rains stop early. They have groundnut rosette disease and leaf spot resistance, and many of the things that the traits that are bred for through many of the strains are including, aside from the resistance and the seed colors, would be ease of shelling, ease of harvesting, and flavor, oil content, things like that generally. There's three here that I'm pretty excited about that are being developed, and they are the improved Rudu, Guerinut, and Acoli White. These are land races. Now, the aspect about land races that are important is that they're locally adapted traditional varieties that have been developed over time through adaptation of natural and cultural, environmental, agricultural practices, and partially due to the fact they've been somewhat isolated. But the consumers and farmers to some extent still prefer land races in a lot of situations. This is what they're used to. This provides the flavor, texture, contents that are important to the consumers. And the other thing is that they are acceptable. Now, one of the problems in generating new germplasm is getting acceptance to these new products. And it takes a while to get the farmers and the consumers to buy in. So improving the land races would suggest that the farmers and the consumers might buy in quicker, especially the consumers, to these land races that have been improved, and these three have. None previously had GRD or LLS resistance. These improved varieties will have, and they will have maturation dates, which is typical of what they normally have in the range of 90, 85 to 100 days. And some additional things like ease of harvest, ease of shelling. So these are going to be really important as they come along for acceptance value by consumers. Alternative host of GRD. This is some recent work we've been doing, which I thought is important and you might be interested in. GRD is endemic to Sub-Saharan Africa, obviously. It was introduced numerous years ago, but the disease is not, since groundnut originated in South America, but the disease is not in South America, it's only in Sub-Saharan Africa. So you had to have had alternative hosts for the disease complex in SSA. Controlling alternative host could be important in an IPM program for GRD, and none have ever been identified in the wild in SSA. Recently, a number of potential alternative hosts have been identified by work that we're doing, and one of those is Senna obtusifolia, which is sickle pod, which is a very common disease worldwide. Indeed, there's a manuscript or paper that's been published years ago and describing sickle pod as probably the most important disease in the state of Georgia. So it's possible that alternative hosts for GRD are worldwide. The confirmation of these potential alternative hosts is in progress, along with their ability to act as viral reservoirs for the groundnut rosette disease. And Henry will be talking about some of this in an upcoming presentation he has entitled Identifying Alternative Hosts of Groundnut Rosette Disease and Virus Complexes. Some concluding remarks then. Little is known about the molecular characteristics of the disease agents, and this is that there's very little known about host pathogen interactions. And this is an area that there really needs to be more work put into in the future. The most intriguing thing is the satellite and the symptoms that it causes and how that works is just really interesting to me, and it's one of those things that I think would provide a lot of information about learning not only more about groundnut rosette disease, but how to control the disease in the future. Very little known, very poor understanding of the epidemiology, both at the basic and applied levels. There's little information known about the alternative host, although I mentioned that. The vector is worldwide. Not much information known about the interaction between the virus and the complex and the vector, nor the biotypes. Are biotypes important in transmission? Certain biotypes important to transmission to groundnut, others elsewhere. I will say that you saw the aphids earlier, a picture of the aphids. You can go into a peanut field, groundnut field in Uganda and just see groundnut plants covered with aphids, as well as some weeds around the field, one of them being senna cyclopod. So there's much work needs to be done on that. Limited genetic resistance is available, but little information is known about the mechanism, although there are suggestions that out there from previous work that there may be other resistances to GRV than those described, as well as there also apparently is resistance to GRAV, but it's not been used today. There is no known resistance to GRD in groundnut germplasm grown outside of SSA. So you have the aphid world, the vector worldwide. You may have alternative hosts worldwide, but any resistance to GRD is in groundnut germplasm only grown within SSA. So there are potential problems there in the future. Groundnut rosette resistance is presently being mapped by Josh Clevenger and at Hudson Alpha Research Institute for Biotechnology and his collaborators. And this could give much needed opportunity for marker assisted selection down the road to speed up programs of breeding. There's a couple of other aspects just to mention. There's frequent dual occurrence of groundnut rosette disease in late leaf spots. So one is left wondering if there's some shared genetics in the resistance between GRD and leaf spot resistance. And groundnut rosette disease and leaf spot resistances have been identified now in the African core collection, which may mean new pools of resistant germplasm are available. And so with that, I'll stop then and take questions that you might have on groundnut rosette disease.